Welcome to our channel, Medico Mentor. In this video, let's discuss CAR T-cell therapy, a revolutionary approach to cancer treatment. We begin with a real-life story that highlights the potential of this therapy. Meet Doug Olson, who was diagnosed with chronic lymphocytic leukemia, or CLL, in 1996. After years of chemotherapy, his cancer was still not under control by 2010. Doug sought an experimental cancer treatment, CAR T-cell therapy. This process involves genetically modifying a patient's T-cells to recognize and attack cancer. Doug participated in a clinical trial to receive this experimental treatment. The treatment resulted in leukemic cells disappearing from his blood, as illustrated by these images before and after the therapy. The initial success led to further clinical trials. In 2017, the FDA approved the first T-cell therapy, called Kymriah. Years later, in 2020, studies showed the modified immune cells were still active in Doug's body, killing any cancer cells that reappeared. His doctors considered his cancer essentially cured. Now that we've seen the impact, let's explore the science behind it. What exactly is a CAR T-cell? To understand CAR T-cells, we must first review how regular T-cells get activated during an infection. T-cell activation is a tightly regulated process that requires two distinct signals. Signal 1 occurs when the T-cell receptor binds to a specific antigen presented by an MHC molecule on an antigen-presenting cell, such as a dendritic cell. Signal 2, also known as co-stimulation, involves other molecules, such as CD28 on the T-cell binding to B7 on the antigen-presenting cell. Both signals are necessary for full activation. When both signals are received, the T-cell becomes activated. Scientists recognized that T-cells' ability to recognize specific antigens could be harnessed to target cancer cells. After years of research, scientists developed the CAR T-cell, a genetically engineered version of a normal T-cell. CAR stands for chimeric antigen receptor. The term chimera refers to a mythological Greek monster composed of parts from different animals, reflecting the hybrid nature of this receptor. The chimeric antigen receptor is a synthetic molecule with multiple domains: an extracellular antigen binding domain, a hinge, a transmembrane domain, and an intracellular signaling domain. The extracellular antigen binding domain is derived from an antibody, specifically the single-chain variable fragment, or scFv, which allows the CAR T-cell to bind directly to a cancer antigen. The transmembrane domain anchors the receptor to the T-cell membrane. The intracellular signaling domains integrate the signals needed for T-cell activation. Importantly, CAR T-cells bypass the need for an MHC molecule to present the antigen, requiring only a single engagement for activation. Because the entire receptor is a protein, it can be engineered by splicing together the DNA sequences that code for its various components. This genetic blueprint is then introduced into T-cells, typically using a deactivated virus as a delivery vehicle. Ongoing research continues to refine CAR T-cell technology, focusing on increasing effectiveness and reducing side effects. CAR T-cell therapy has evolved through several generations, primarily distinguished by the number of signaling domains tucked inside the cell. While the first generation featured only a basic activation trigger, later versions added boosters to help the T-cells live longer and fight harder. Recent advancements include the approval of new therapies, such as Aucatzyl in November 2024, for adult patients with relapsed or refractory B-cell precursor ALL. Like any powerful treatment, CAR T-cell therapy has potential side effects. When CAR T-cells engage their targets, they can trigger a massive immune response that prompts other cells to release signaling molecules known as cytokines. While this is a sign the treatment is working, an excessive release can lead to cytokine release syndrome, CRS. This typically manifests as a mild fever, but can, in rare cases, escalate to life-threatening organ failure. Additionally, these cytokines may compromise the blood-brain barrier, potentially causing immune effector cell-associated neurotoxicity syndrome, ICANS, which can lead to severe neurological complications. What does the future hold for CAR T-cell therapy? To increase precision, researchers have developed tandem CARs, which are engineered to recognize two distinct proteins. The T-cell is only fully activated when both targets are detected, reducing the risk of attacking healthy tissue. For the more difficult challenge of solid tumors, which often use off switches to deactivate the immune system, scientists are creating armored CAR T-cells. These cells are genetically modified to be resistant to these inhibitory signals, allowing them to remain active within the harsh tumor environment. Finally, to improve safety, self-destruct CAR T cells, often called suicide switches, include a specific protein that triggers cell death when a particular drug is administered. This gives doctors an emergency kill switch to instantly stop the therapy if side effects become unmanageable. Other future directions include developing, quote, off-the-shelf allogeneic CAR T cells, which could be readily available for more patients. To learn more, please explore the suggested resources, including videos and articles listed here. Thank you for listening. Subscribe to our channel Medico Mentor for more such videos.